Bueno, muchas gracias por venir, por asistir. Hoy tenemos el primer ciclo de conferencias del 2020, el cuarto ciclo, y vamos a tener el honor de contar con la doctora Chiara Batini, quien es investigadora de la Universidad de Leicester y quien amablemente ha cedido su tiempo para hablarnos sobre estudios de asociación de genomas en grupos multiétnicos. Realmente es una investigadora muy importante en su campo y nos conocimos hace un par de años en un curso de herramientas informáticas y bueno, la idea es compartir su trabajo y cómo ella nos ha usado las herramientas informáticas para hacer este tipo de estudios más allá de la parte genómica que se realiza. Sin más preámbulos y en vista del tiempo, procedemos a iniciar la conferencia. ¿Puede?
¿Ya?
Ya.
Muchas gracias, Rafael, por la invitación y buenos días a todo el mundo. Solo quiero decir que sí hablo español y me pueden hacer preguntas en español si quieren, pero me resulta casi imposible dar la charla en español, entonces perdón, pero será en inglés. Y vale, ahora han desaparecido, pero bueno.
Apagamos el video para mejorar la conexión.
¿Quieres que lo apague yo también?
Sí.
¿Está apagado? Sí, ¿no?
Sí, sí.
Vale, muy bien. Entonces, I'll switch to English now. I'm not a native speaker, so hopefully I'll speak clearly and slowly enough. Please do interrupt me and ask questions at any time. So I have agreed with Rafael to talk about multi-ethnic genome-wide association studies for this seminar. I will give you an overview of the talk in a second. You will see that maybe the topics are a little bit wider than that, but I hope it will still be useful and generate some discussion. So I'll first would like to, wait, there you go. I would first like to introduce myself. So I'll give you a background of who I am. I have a background in biological sciences. I've done my university studies in Italy, Rome, where I'm from. And then I've done my PhD in collaboration with an institute in Barcelona. And in that period, I was working on the ancient peopling of Central Africa. And when I say that, I talk about human populations. And then I moved to Leicester about 10 years ago. I spent the first 6 years of my life here working in the Department of Genetics, working still on human population genetics, but in Europe. And then a few years back, I've moved to the Department of Health Sciences, where I am now. And with a series of postdoc and fellowships, I'm now working on the genetics of smoking behavior in non-European populations. So when I thought about what to talk about exactly, I thought that in order to talk about multi-ethnic genome-wide association studies, I would have given you a little bit of an introduction on human genetic variation and what it is and how we got where we are and how it is detected. And then a brief introduction about genome-wide association studies in general, what they are and what they have achieved so far. I will have a section about trans-ethnic and multi-ethnic genome-wide association studies, the ethical and technical implications, and then a very light touch on my contribution. So I'll show you a bit my collaborators and what we are aiming to do. And then I've decided to close the seminar with a few slides about the role of bioinformatics in all of these. And I hope that will generate a bit of a discussion about where we are at and how is bioinformatics going to evolve in the next few years. So I'm sure you all are all aware that the human genome is constituted by 46 chromosomes, organized in 23 pairs that are in the nucleus of the cell, one of which is our sex chromosome. And then we also have some genomic material in our mitochondrial DNA. So if we look at comparing human genomes across individuals, or comparing to a reference sequence that is the one here above, we see that we can have different kind of variation among, well, it's not only in humans really. So you can have sequence level variation and structural variation. At the sequence level, we have single nucleotide variants or polymorphisms or SNPs, which are the substitution of one specific nucleotide at one position. We have short insertions and deletions, so that will involve a few base pairs. And then we have a number of structural variants that are include, they're basically chromosomal rearrangements, which I'm not going to go into detail because I'm not really going to talk about structural variants pretty much at all. So let's try and give some numbers to this. So the human genome is 3.2 billion base pairs long. Among human beings across the globe, there have been more than 100 million SNPs have been described to date. We have about 3.6 million short indels described and about 60,000 structural variants. The numbers in these slides come from the latest study by the 1000 Genome Consortium, which I will show you again in a second. Some of them may not be completely up to date because this was published a few years back, but the SNPs figure, I've updated it with the latest references. So I'll give you a very quick history of how we got where we are. So I think this story starts when in 1953, DNA structure was defined by these people that you've probably heard a lot about already. And then there were two important technological innovations that happened about 30 years later. So we have the possibility of sequencing DNA with the Sanger sequencing method published in 1977. And then the possibility of making this automatic with the PCR method that was published in 1983. And it's not long after that that genomics as a discipline is actually defined and the Genomics Journal is founded. And again, not long after that is when the Human Genome Project is launched. So this is in 1990. And after the Human Genome Project has been launched, in the following years, in the following 10 years, there have been the complete sequencing of the genome of several organisms, including bacteria, yeast, insects, and also more complex organisms such as chimpanzee. So in 2001, there was the first draft of the Human Genome Project, which was officially completed in 2003. And in 2002, we have the launch of the International HapMap project. So this project was particularly interested in understanding the genomic structure and particularly the extent of the haplotypes among human beings and specifically across continents. So it did select a population in Africa, in Asia, and in Europe, well, representative of, I would say, rather than in there. And they basically did a very dense genotyping of these individuals to understand how the haplotypes are organized in different populations and how demographic history affected this. After this, we have the launch of several other projects, some of them specifically focused on more functional aspects. We have a focus on cancer, on the microbiome, but I think the most important in that list for this talk is the 1000 Genome Project, that project that was launched in 2008, and is pretty much a natural continuation of what HapMap was doing. So it includes several populations across the globe, and what they have done, they have sequenced their whole genome at a low coverage. And for some of them, they've done some high coverage sequencing in order to characterize all the extant variation in these populations. These two projects are completed in the 2000s, so in 2010, 2015, so really not long ago, if you think about it. And since then, we have seen the launch of several larger scale projects, mostly aimed at understanding clinical phenotypes better. So we have examples such as Genomics England, which is a project that includes about 100,000 individuals in the UK, mostly selected on the basis of their clinical phenotypes and with a strong focus on rare diseases and cancer. There is the Precision Medicine Initiative in the US. There is, not included in this list, UK Biobank, which I'm going to talk about later, which is a cohort of 500,000 individuals for which there is dense genotyping data, and there is already some sequencing data, and there will be in the next 5 years whole genome sequence for all of them. So large scale efforts. So this is a very, very rapidly evolving story towards the recent times. And what have we achieved? So I think the first contribution that human genomics has given to science, but also more in general to society, is represented in this slide. And I'm gonna guide you through it for those who are not so familiar with the 1000 Genome Project. So here you have all the populations that have been included in the project in the world. The size of the pie chart is proportional to the number of variants that have been described within each population. So you see these pies are bigger in African populations, where there is most of the genetic diversity described. And then they are colored in two main shades, let's say. So there are some gray shades that represent the proportion of variants that are shared across at least 2 continents. And then the colored shades are the variants that are private either to the population or to the continent. So I think the first big observation is that most of the variation is shared across continents. And then the slice that represents the variation that is private to the population is generally the smaller slice in all populations consistently. So I think the main message here is we're all the same with some differences, on average at 3 million SNPs. And this was really important because it was really the yet another confirmation that you can't really talk about races among human beings. Of course, these differences is what we are interested in, is what you look at to understand demographic history, and also what you look at to understand why different people are more have different risk of diseases or present different behaviors. So this is what really we work on. So if we take that timeline that I was showing you before, so this is compacted, what happened to the technology at the same time? So I really see that what happened is a drastic change that happened in the early 2000s. So we have Sanger sequencing, restriction fragment length polymorphisms, and back and cloning being the main technologies before 2000, and then microarrays and next generation sequencing taking the lead after that. So very low throughput technologies and then very high throughput technologies afterwards. So I'm going to focus on the high throughput technology and just have a very brief look at how they differ. So SNP microarrays, of course, they look at specific variant sites in the genome, while next generation sequencing is a massive parallel method of sequencing genomes in small fragments and then reassemble them or realign them through analysis afterwards. One main difference is that the first are relatively cheap per sample. The second is relatively expensive per sample with the same, when you reach the same quality of data. The kind of analysis is in terms of just processing the raw data analysis is a lot easier in the first case. You need a lot more bioinformatics support to analyze next generation sequencing data. You have some biases in the first case, as in you need to know the variants you want to look at, and you can only look at a certain number of variants. Still, this number can be very high as 5 million at the moment. But there is a bias towards variants that are more common in European populations, even though there has been some efforts in overcoming these. There is no real bias in next generation sequencing, even though technically there is still a bias towards the reference allele, which is most common in European population, but it's a much, much smaller bias. So thanks to the introduction of microarrays, I think that's the technology that really pumped the beginning of the genome-wide association studies world. So it is really maybe 13 years that we have genome-wide association studies, and they're becoming more and more popular and larger and larger. The idea is fairly simple. So these are hypothesis-free methods that are used to identify an association between a genetic region, so a SNP or an indel or a locus, and a trait. And this trait can be a disease or it can be a behavior. They have been really applied to almost everything you can think of. They are not specific to human populations. They're very much being used in plant biology, for example, but they're used in other species as well. Is a statistical analysis. You basically check if there is an association, and if that association is significant, your allele is associated with the trait. So I will do a toy example. I wasn't too sure how many people really captured that concept very quickly, but this is a very small toy example. So you want to start with, so let's think about cases and control study design. Not all the studies are designed in this way, but you would like ideally to have your cases and controls coming from the same population. And when I say population, I don't only mean where people live or where they're from or their genetic ancestry, but I also mean similar demographic characteristics in terms of age and any other characteristic that you're not really studying. So sex ratio, anything that is not the interest of your study. Ideally, it should be comparable between the two groups. So then you compare at different SNPs. So I've done here 2 toy examples. So we're looking at 8 individuals per population in the cases and 8 in the controls. And you basically check how the allele frequency compares between the 2 groups. So this is so in the first case, there's no significant difference. In the second case, the allele G is overrepresented in the cases. So you could say that G is associated with your trait. Now, this is a very simplistic view. Of course, the models are a lot more sophisticated than that. And you also can use you have to use different models depending on which kind of phenotypes you're looking at. So if you're looking at as a case control study design, so a binary phenotype, or you're looking at something that varies quantitatively. Linear logistic regression models are used very often. They're not the only ones. There are other models that can be used, but the good thing about regression models is that you can incorporate covariates such as age, sex, and anything you think that co-varies with the phenotype. So you do this across the genome. So you've seen 2 SNPs before. Here you've got 43 million SNPs that have been included in this analysis. The way that the results are represented are in the form of a Manhattan plot. So on the x-axis, you've got the genomic coordinate organized by chromosome. And on the y-axis is the minus log 10 of the p-value. So the higher the higher the dot, the lower the p-value. And then that threshold that you see there is the Bonferroni corrected significant threshold. So of course, in these studies, you're testing many variants at the same time, massive issues with multiple testing. What it has been estimated, but what you're interested in here is how many independent tests you're running on the human genome. And it has been estimated that this is about 1 million. So this corresponds, because we know that there is a lot of correlation between variants in the human genome because of linkage disequilibrium. So this corresponds to basically the number of independent haplotypes that you have in the human genome. Of course, you may or may not have enough power to detect your real associations, and your statistical power will depend on a number of things. Sample size is really important. The effect and the frequency, the effect size and the frequency of the variants you're looking at also are really important. So the stronger the effect, the easier it is to detect, unsurprisingly. The more common the variant is, the easier the association is to detect. But also it depends on the linkage disequilibrium between the variants that you are analyzing and the causal variant. So this means that this basically refers to the fact that if you are using genotype data and not sequencing data, it's very unlikely that you're actually including the causal variant in the variants you're looking at. So all you are relying on is on variants that are linked or correlated to the causal variant you're interested in. So depending on that, your power may be higher or lower. And we'll get back to this later when we look at the trans-ethnic side of things. There are, of course, potential sources of false association. Genotyping error is a very, I guess, obvious one. Population structure is something you really need to take care of when you run GWAS, and relatedness between individuals and rare variants can also be an issue. Some of these may be mitigated depending on the model you decide to test your association with. Population structure, I will give you a very silly example that might have been an issue initially, but is thank God not an issue anymore. So it's very easy to understand if you assume, for example, that you collect your cases from one population and your controls from a different population. When you're going to test for differences in allele frequencies between the two, you're going to pick up also any allele that has differences in allele frequencies because of demographic history and not because of what you're actually comparing. So you need to take care of that issue. There is a more fine level of population structure that you need to take care of. Usually the way this is done is by performing a principal component analysis. That is what you see there on the right. Define your sample according to the mean principal component values of principal component 1 and 2 and their standard deviations, and then use the values of the first 5, 10, 15 principal components to correct as covariates in your model. So take care as much as possible of the relationships between the individuals that way. There are other ways to do this. We can talk about it later if you're interested. Anyway, FAQC is, of course, very important. And that is usually where 90% of the time goes into when you're doing this analysis. So what they have achieved so far. So this is a picture that comes from the GWAS catalog. It summarizes the results of more than 4,000 publications and more than 175,000 unique associations described to date. The way you need to look at these is that each of these blurred, can you see my mouse? Yeah. So each of these is a chromosome, which is here, and then you have little dots colored according to which class of phenotype you're looking at. So there are many associations described, a lot less in proportion on the X chromosome and none for the Y chromosome. And if you're interested in what kind of traits people have looked at, this is a very quick picture of the groups that are represented here. So you really have all sorts of traits that have been analyzed. And of course, Rome wasn't built in a day. So this is a picture, it's the same picture we were looking at before, but maybe with a bit less data because it's updated to 2016, but you could imagine this is a trend that will keep growing. So here you have time on the x-axis and number of cumulative SNPs discovered on the y-axis. And for each year, you have basically how many SNPs have been discovered for each trait, and then so the bars are colored according to the classes. So what you see, basically, the main message here is there are more and more SNPs described each year. There are more phenotypes analyzed each year. So it is becoming more complex. It is becoming more informative. So I'm sorry for the boring slide, but a bit of a summary of what they have achieved. So GWAS have taught us that complex traits are highly polygenic. So you have many variants that contribute, many genetic variants that contribute to explain the phenotypic variants. And each variant only contributes a little, really. And that's why you need larger and larger sample sizes to actually discover the associated variants. And of course, each of us will carry a number of alleles that either increase or decrease the trait of disease. So there will be unique combinations of these in each individual. Pleiotropy is pervasive. So you have in many cases, the same variant can be associated with different diseases and traits. And you have genetic correlations among different traits. And then you have something that has become more and more popular, and for many people is a real hope for the future, is the polygenic risk scores. So using the information from GWAS and by combining the effect sizes described for genetic variants and the number of alleles carried by each individual. So if you combine these two information and you sum them up all together, you can come up with a polygenic risk score that can tell you the risk of that disease or that trait for a specific individual. Now, at the individual level, there are still a number of issues in using them for prediction, but they have become quite useful when you need to define groups. So define like a group of individuals at higher or lower risk in the population, and also in Mendelian randomization analysis. Of course, along all these, there has been a lot of method development. Data sharing has been key for achieving all of these, and I'll get back to this at the end. And also, what has become really important in later years is the availability of omics data. So other kind of other levels of the biology that have allowed the functional follow-up to analysis to be performed at scale. So what you can do is looking at gene expression data, is looking at epigenetic marks, and you can actually move forward from, okay, this variant is associated with this. What does it actually mean? And it has been shown that if you have support from genetics in identifying a drug target, the drug targets that have this support have a much higher probability of making it into phase 3 trial or into the market when tested. So these are some examples. I'm not an expert in all of them, but I think this is becoming more and more really the focus of what people and industries really are interested in. So there are now dedicated conferences to these. That is, so for example, one quite important one is the target validation organized by the EMBL. Okay, so I'm now moving towards the multi-ethnic or trans-ethnic genome-wide association studies. Sorry, the acronym is misspelled there. There should be another S. So I'll first give you a picture of what the situation is. So this, what you have here, you have 3 pies. So we're going to start with the one top left. So that represents the ancestry of the individuals included in the studies. So you see that there is a huge bias towards European individuals. This is probably not a surprise to you, as it has been in the news quite a lot lately, actually. There has been some effort in representing Asian individuals, but all other ancestries are very much underrepresented. If you look at the same picture, but focusing on the studies, you see that there are quite a lot of studies that actually include multiple ancestries, including Europeans. So that bias seems to be a little bit reduced. But I think the most interesting part of this slide is bottom right, so the associations. This tells you the proportion of association described when including individuals of that ancestry. And you see that, for example, there are about 2.4 African individuals included in the studies, but 7% of the associations are described when analyzing those individuals. So this seems to suggest that there is a disproportion in the associations you can actually discover when you look beyond the European genomes. This is another way to look at the same picture as before. So this is giving you a time perspective. So I think it's pretty much the same data. So you have time on x-axis, and you have the number of individuals in the top plot and the fraction, the proportion in the bottom plot. So what you see is that, yes, there is an attempt in later years, but that is not nearly enough to be representative of the global population. So if you compare this proportion to the actual proportion of people that live in the world, there is no way near an equal representation of human beings, which of course can exacerbate incredibly the inequalities that already exist for many other reasons, but you know, health inequalities are part of it too. Okay, so I'm going to move towards technical implications of including different populations in a GWAS. So let's focus on the plots on the left first. So here on the bottom, you've got the SNPs, right? And your square one is your causal variant. So we are in a situation in which we are not looking at our causal variant as such, but we are actually only looking at A, B, C, and D. Okay, so these are SNPs that are in various linkage disequilibrium with the variant. The level of linkage disequilibrium as measured by R square is here. So the higher this number, the stronger the linkage is. So in population 1, variant B is in very strong linkage disequilibrium with the causal variant. And as a consequence of that, the p-value is lower in this variant. Okay, so we do observe these, but we don't know what is representing exactly. When we look at population 2, B is no longer in LD, but D is in strong LD. So we see a similar result, but on a different SNP. So depending on which population you're looking at, you may or not be able to detect the association in the first instance. And then, of course, depending on which comparison you do, you may or may not be able to see a concordance in the effect that the variant has on that trait or phenotype. And that is very important when we look at, it's very relevant to fine mapping. So fine mapping is that process in which, so we have all this data, we have these very dense genotypes, we have imputation data. So out of genotype data, we can impute genotypes at other sites. At the moment, you have panels that include as many as 90 million variants that you can have information on with different certainty, but still very useful for analysis. But we want to know what is the biology behind all of this, and that starts from understanding what the causal variant is. So here you have pretty much the same as before. So you have, this is a genomic region, okay? So imagine these are genomic coordinates on the x-axis. The LD strength is represented by these colors. And what you see is that if you test the phenotype in population 1, this is your the SNP with the lowest p-value. And a number of SNPs in LD with it also will show you a signal. And that the same happens in this other population, but there are two different SNPs in two slightly different positions. So if you then try and find map, what you can see is that the higher probability for your causal variant is going to be at the intersection of these two intervals. So by including populations that have very different LD patterns, so the length of the blocks is very different between population, you can actually nail down the causal variant a lot better. Not necessarily you will be able to, but you have better chances. And in this context, because LD blocks depend on the demographic history of the population, and because African individuals, so because we have originated in Africa, and that's where the most recombination has happened really, because we have been there for longer. So that's where haplotype blocks are the shorter, and so they're particularly, that's probably why they're particularly useful and valuable to the study of, to genome-wide association studies. Okay. And then there is another issue. So demographic history has an impact on LD, but it also has an impact on the distribution of alleles. So this is a very schematic view of that. So we originated in Africa, expanded out of Africa. And while we expanded out of Africa, we only brought with us, because of a bottleneck, just part of the genetic variation. And that continued to be the case also in the Americas, which I'm really sorry are not represented in this picture. So that means that different alleles will have different frequencies. And we have said before that the allele frequency is one of the factors that will impact on your statistical power in your association testing. So this means that by studying different populations, you may have different power in actually detecting even shared associations. And both these aspects are relevant to the polygenic risk scores. So going back to polygenic risk scores, what you have here is the prediction accuracy. So, okay, what you have here is, so you have your GWAS, which has been run in a European population, and then you test the prediction accuracy of the polygenic risk scores built on the results of that GWAS in different populations. And you see that this accuracy is inevitably higher, one, in Europeans, but it decreases in other populations, being the lowest in African populations, mostly because of what we have discussed before. Okay, so in this context, it's clear that we need to do something about it. I think there are a lot of people that are doing their best. There are some large biobanks that have been created in different parts of the world, China and Japan in Asia. We have H3 Africa in Africa, the Million Veteran Program in the US, TopMed is a multi-ethnic cohort in the US. They have different degrees of accessibility, but these are important projects that are happening. But of course, there are more smaller scale projects that are happening as well. So I'm going to now give you a bit of an overview of my collaborations and what populations I'm working on. Of course, I'm interested in smoking behavior. The reason for that is mostly because smoking is one of the main risk factors of at least the top 5 causes of disease, of death globally. And the perception of smoking has changed dramatically in the last 60 years. So what you see on the left are advertisements of cigarettes in the 50s, and the messages that Public Health England is sending out today. So it's really quite different what you see. And also, of course, smoking is perceived very differently across different cultures. I come from Italy. Smoking in Italy is different from what it means smoking in the UK. So it really is can be quite granular. So also, the number of smokers varies among the different world regions and among different countries. It also varies dramatically among genders. So males and females smoke very different prevalence. Generally speaking, women smoke a lot less than men in the world, with some important exceptions such as Europe and the US. Of the 1 billion smokers in the world, 80% live in low and middle income countries. Smoking prevalence is expected to increase in 2 specific regions in the world, according to predictions from the World Health Organizations. And these are Africa and the Middle East. So I've started to tackle these, but so basically I decided, okay, well, let's look. So we have a lot of data on smoking behavior, on the genetics of smoking behavior in European populations. I think they've reached up to 1 million individuals. So the latest GWAS has included 1.2 million individuals from Europeans. So I started looking at non-European individuals in UK Biobank. So if you're thinking this is a population of the UK, yes, you're right. This is 500,000 people that live in the UK. But the good thing about the UK is that it's quite multicultural. So forgive the terrible tags that the census gives to define different ancestries, but that's what they are. But what you see is that there is a representation of non-white individuals in the UK. Even though this is not proportional to the actual number of individuals that live in the UK, is still quite interesting sample sizes, considering that they are not from the original countries. So for these individuals, there are genotype data. There is a specific UK Biobank microarray. There are 90 million imputed variants, and there are nowadays 50,000 available whole exome sequences, and within the next 5 years there should be exome sequences and whole genome sequences for the whole 500,000. So the NHS is very much interested in the UK is very much interested in also expanding beyond this, and they have a plan of sequencing about 1 million individuals in the next 5 years. So there's a lot of interest. We may discuss later what is the value of having all these different levels of genotypes. I mean, why would you not stop at the whole exome, or why wouldn't you skip it if you're doing whole genome? I think there are different starts really behind it, but anyway. There is intense phenotyping on them, and also there is linked electronic health records available for half the cohort, and it will be available for all of them soon enough. So I'm looking at the African individuals and the Asian individuals in the UK Biobank, and I'm also collaborating with other cohorts. So one specific one is the Awijan cohort in Africa. This includes 12,000 individuals and is led by Michelle Ramsey in Johannesburg. It includes individuals from 4 different countries. It's a massive effort they have done there. I feel very privileged that I can collaborate with them on this. Another project that it has a much slower start, but hopefully in the next 10 years we will see something out of it. We have put together a consortium in Africa to evaluate strategies to stop smoking. From what I have understood, African individuals tend to smoke a lot less than European individuals, but they quit. It's harder for them to quit. So there's something about how you approach the cessation strategy that really needs to be thought in a different way. And also, we really don't have any data at all on cessation strategies in the continent. Well, we have very few, let's say, and the only clinic for cessation that we have found is in Cape Town and is led by Richard van Zyl-Smit, who was the director of the Lung Institute in Cape Town. I've also very recently, so another aspect of all of these is that admixed individuals tend to be excluded from analysis very often because it's too difficult to analyze them. But I've recently signed a data agreement with the Mexican Biobank, and I'm collaborating directly with Andrés Moreno Estrada and Mashal Sohail at Langevio to include the Mexican individuals in this work, and especially to build a pipeline that can be used to analyze admixed individuals in general. So also all those that come from other larger cohort efforts. And then a very small contribution, but I thought I'll mention it because it might be a study that is of interest to some of you. So we have a local cohort that is called Exceed and is led by my supervisor, Martin Tobin. This includes about 10,000 participants to date, and only very few of them have South Asian ancestry. This is very much in disagreement with the actual demographics of the area, where we have one of the largest populations, South Asian populations in the country, and in Leicester, there is about 30% of South Asians in the population of Leicester. So it's quite a big proportion of the local population, definitely not represented in these numbers. So we are now trying to develop a different culture-aware strategy to involve people in different ways, really. Okay, so I'm sorry if I haven't presented you any results, but I thought I'll use the last 10 minutes to actually bring things back to the bioinformatics perspective. As you probably have guessed, I really enjoy timelines. So I've done timelines for bioinformatics as well, and I'd be very interested to hear what you think about that at the end of the talk. Okay, so from what I could gather, bioinformatics really starts in the late 50s, well, early 60s is the first bioinformatics software. And the initial need for bioinformatics was analysis of protein sequence, because proteins started to be sequenced before DNA. So we have the first software in the early 60s, and then the first biological sequence databases, which was an atlas of protein sequence and structure. And then in the 70s, 80s, we have publications of softwares to analyze sugar sequences. We have some very classic phylogenetics and multiple alignment software, such as Philip and Clustal in the 80s. I didn't know Clustal was actually so recent, really. And then we also have some proprietary softwares that were developed in these years to kind of meet the need for analyzing and handling DNA, RNA, and protein sequence data. But I think one big step in bioinformatics is something that happened more in general, I would say in computer science probably, which is the foundation of the Free Software Foundation. So that's the beginning of open source software, and is the publication of the GNU Manifesto. Sorry, I needed to drink. So this is a very famous quote from Richard Stallman, which is the leader of this movement, really. So we're talking here about free software, but this is a matter of liberty, not of price. To understand the concept, you should think of free as in free speech and not in free beer. So this is not about software for free, but this is about software that can be run, copied, distributed, studied, changed, and improved by others and redistributed around. So it's a whole cultural change, really. And I think this really prompted it was really helpful to bioinformatics, because if you think about it, most of the software is open source. There is a lot of sharing and a lot of contributions to different pieces of software, and that is fantastic. I think it's a great power that we have nowadays. Just out of curiosity, I want to check when Creative Commons was launched, and that was much later. Actually, it was in the early 2000. And then, with great surprise, I found out today that GitHub was developed in 2008, but it was acquired by Microsoft in 2018, and that really surprised me. Then in the mid-80s, another big important step that happens is the standardization of data formats, the definition of essential metadata for sequences, and the facilitation of data sharing, especially between these three big databases. So we have GenBank, EMBL, and DNA Data Bank of Japan. So they start to have some common definitions, and that really helped in the sharing of data among researchers, really, ultimately. And then we have the Foundation of Bioinformatics, which initially, so this is the journal, and initially it was called Computer Applications in the Biosciences. And the mission of this journal was to democratize bioinformatics among biologists. And in their mission statement, they say that computing should be no more mystical a technique than any other laboratory method. And if you ask me, we were definitely not there in 1985. Probably we were not there when I started university, and I wonder how close we are to this now. I think we are a lot closer, but I bet there are still a lot of biologists that see bioinformatics as something hard and difficult, and I don't want to know about it. So yeah, so that's I think something that we need to think about a bit more. Perl and Python are created in the late 80s. Perl is the lingua franca of bioinformatics pretty much until the late 2000s, and then Python takes over. So what you have nowadays is that depending on when you started, you will be great in one or the other, or in both if you're really good, I guess. I really don't know Python, but everybody tells me that it's a lot easier than Perl, and it's a lot more straightforward, and that is really the power over Perl. But both of them have modules and libraries that are specific for biological data. So, you know, they are really the preferred languages. But then, of course, a big step forward. Comes online. Hello. Yay.
Regresamos.
Yeah.
Lo del Excel. Le da desinstalar, ¿cierto? Ok, ya. Yes. We are online.
Okay, so shall I go?
Yes.
Okay, I only have 3 slides left, and then I hope we'll have a discussion. So I'll just finish these. So I was just saying that, of course, things changed a lot when the World Wide Web was invented. So databases were made available online. You have bioinformatic tools that become available, some of them with a graphical interface, some more user-friendly, trying to kind of cope with that fear of the command line that most biologists have. And then you have data and publications that are shared across the globe. So lots changes then. Wait, where am I? Where is my mouse? Okay. And then in 2000, we have large projects from, the bottom part of the timeline. Technology changes. So we have microarrays and NGS data. So lots and lots of data on many, many people. But at the same time, we have high-performing computing technologies that have matured with time. So you have massive parallel computing that allows genomics to basically analyze the data. And storage and computing resources become ever so slightly more accessible. There is a significant increase in user-friendly tools and especially in online forums for help and advice. And I'm thinking of Seek Answers and BioStar that I think are brilliant or Stack Overflow, all brilliant resources. Also, bioinformatics becomes part of university curricula. So that is really a key change so for those of you who are my age, you're probably those that started when bioinformatics didn't exist and then, moved into this. And then some of you will be younger and started as bioinformatician. So there is a whole diversity about how people arrived into the field. But the need for bioinformaticians' jobs explodes, really. So bioinformaticians are the most requested for many years when all this data is produced. So you have people that move from biology into bioinformatics, people that move from computer science into bioinformatics. So it's really quite a diverse set. So I just wanted to conclude with a few reflections that I've thought about. Some of them I've read, some of them I thought about myself. So one of them is something I've read, and it was about why is biology the only discipline that has a specific term for referring to the use of computers. So you don't have you have bioinformaticians in biology, but in physics and mathematics, they just use computers. It's just part of the discipline. And is this actually changing? So do we actually need to talk about bioinformatics? Or is this now biology and that's it? That is just part of what biology is. I think we are going to face big issues in terms of reproducibility of pipelines, data integration, and data complexity is increasing. So I'm thinking very much about clinical data, electronic health records, and the type of data you have in there. So it's becoming more and more complex to actually extract information from these. And then I wanted to go back to the point about democratization. Is this actually happening? Is bioinformatics making things more democratic across the globe? So I invest a fair amount of my time in training and capacity building in that way. I think these things are happening. I think there's more and more option, but it's still very biased depending on where you are. But also bioinformatics comes at a cost. So I think it'd be interesting to think, how can we make this sustainable? how can we actually make it democratic? So I think these are things that are not really resolved. There is a very, very fast pace in some parts of the world, not the same everywhere. These are issues that I think we should tackle. And I think I'm going to end by just saying thanks to the team where I'm from. So I work in the Wayne Tobin team. Nick Shrine and Martin Tobin really shaped the way that I've moved into this discipline, and Nick especially shaped very much the way in which I do bioinformatics today. I still don't consider myself a bioinformatician, even though probably I am, and that's why I was invited. But I think we can talk about how you define yourself for a long time. So thank you very much. I'm really sorry that this was really long. I hope it kind of can prompt some discussion there.
Yes, yes. Thank you so much. Muchas gracias, doctora Chiara. Excellent presentation. ¿Algunas preguntas?
Questions in Spanish or English or Catalan or Portuguese? No problem.
I can't read here, so someone can repeat.
Pregunta. Una pregunta. Por acá tenemos una pregunta.
Okay.
Question.
El micrófono está acá.
Acá. Aquí al lado.
Sí, en la cámara.
Hola.
Hola.
Bueno, yo no he trabajado mucho en bioinformática por el momento, ni en genética, pero bueno, estoy incursionando. Y si hay alguna, o sea, son dudas más generales, pues, porque, como digo, estoy comenzando. Y, por ejemplo, habla sobre variabilidad genética, por supuesto, siempre con respecto a un referente, a una referencia que, bueno, en mi caso desconozco cuál es la naturaleza de esta referencia, y mi pregunta es si esa referencia como que va cambiando en el tiempo, es decir, a medida que aumentan los datos, no se readecua lo que se considera normal o no normal, pero referente con respecto a lo que se va encontrando en las diversas poblaciones. No sé si
Do you mind if I answer in English? Because it's still easier. So I think you make a very good point. So the reference genome is the one that was sequenced in the Human Genome Project. So we still use the same reference genome. Everybody in the world uses the same. It makes things a lot easier that way. It was sequenced out of a mixture of, I think, about 20 individuals originally, with a huge European bias. Okay, so no, it doesn't necessarily represent every human being in the world. There is curation, reference genome, that includes like incorporating the variation. So this is the situation. Nevertheless, however, you do, so despite the fact that you compare to the same, then you have, for example, if you are familiar at all with next generation sequencing, variant calling, in that stuff you have a recalibration usually. And when you have the recalibration, you do incorporate the variants described in the project. So there are other ways. So what I'm trying to say is that there are other ways to incorporate the variation and consider it, but it's not in the reference genome.
Entonces, lo que se considera susceptibilidades o no susceptibilidades de enfermedades, con respecto a los SNPs, por ejemplo, o a cualquier otra variante, es con respecto a esa persona del proyecto Genoma.
So the reference genome is only, no, so you still look at individual genotypes. So for example, if I'm doing, like in UK Biobank, what happens is that you have genotype data. So it's not sequence data, it's genotype data. you have genotype data, you look at the genotype of the individual and you compare among them. So you're reference genome free, effectively. So that is less of an issue. It is a bit more of an issue in terms of next generation sequencing, because you need to realign back to a reference genome. But nevertheless, all the sites that are different, and then you compare across among samples. So it creates a little bias, but you don't bias your observation towards the reference genome completely. So it has a very slight bias in the variant calling process, but it's not massive usually. And it also depends on which method you're actually using. So different variant callers have different biases.
Y la última, que no sé si es políticamente correcta, pero con esos datos de variabilidad de cromosoma Y, ya podemos decir que todos los hombres son iguales.
Sorry, but you have to say it again.
I'm asking if with that data about the Y chromosome, can we say all men are green?
No. No. So what that diagram is telling you is that we didn't find any association on the chromosome, but it's just because it's understudied. And also, so I'll be not politically correct myself and say that it's a bit of a useless chromosome, and I don't mean it in a social way. I just think that it has very few genes. Is actually a little bit of a useless chromosome. I'm sorry, guys. I've studied it very, I love it. I don't have anything against the Y chromosome, but it doesn't have many genes.
Then we have another question.
Sure.
Buenos días, profesora, mucho gusto, muchas gracias por su presentación. Yo trabajo en biología, pero también estudio una bacteria, Helicobacter pylori, que es uno de los principales agentes de cáncer gástrico. Y en Venezuela, en la región de los Andes, tenemos una elevadísima prevalencia de esta infección. ¿Me sigue, profe?
More or less. I mean, you have a little bit of an echo. So you said that I didn't understand what organism you're working with.
Helicobacter pylori.
Oh, yeah.
Well, I can try in English, but
no, it's okay in Spanish. It's more, the problem is more the audio. So if you get closer to the microphone, that might help.
Okay. Esta infección es elevada, muy elevada en la región de los Andes, en Venezuela, así como en Colombia. Y igualmente hay una elevada prevalencia de cáncer gástrico, de las más grandes en el mundo. Sabemos que el cáncer gástrico es multifactorial, ya en esta población las personas también tienen un elevado consumo de alcohol, de chimón, tabaco, una dieta elevada en sausage, productos ahumados, etc. Pero mi pregunta va a probablemente un estudio de variabilidad en esta población, de variabilidad genómica, pudiese ayudar a dilucidar o a identificar si está más relacionado al origen de esta población, que es europea. O sea, principalmente la población de los Andes es de raza blanca y descendiente de europeos, a diferencia de otras poblaciones en Venezuela, como en la costa venezolana, donde también hay una elevada prevalencia de Helicobacter pylori. Y bueno, quizás un estudio de variabilidad genética, ya para las personas especialistas en esta parte epidemiológica, pudiera dar más información sobre esos factores que realmente están definiendo estas elevadas tasas de infección y de cáncer en la población de los Andes. ¿Cuál sería su opinión?
Okay.
Sí, traté de ponerlo en contexto de este proyecto.
First of all, I'm not an expert in Helicobacter pylori, okay? But I do know that you do have co-evolution between humans and Helicobacter. So there are adaptations that humans have and so on. So I can't not answer your question, I think, technically, but I may give you something that might be a bit more valuable. So there is someone at University of Leicester that is working on a project on Helicobacter pylori host parasite coevolution in Cape Verde, and she is Sandra Beleza. Have you ever heard of her?
No, well, I know Dr. Dominguez Bella is one of the.
So I really, because she has collected, I can't remember how many samples, but it was more than 100 samples, and it's paired human and Helicobacter, and she has sequenced the genome of Helicobacter and done the genotyping of humans. So she's got quite a strong data set. And Cape Verde, so you are mentioning that this population has got a European ancestry and wondering if that is increasing the risk factor. So the interesting thing about Cape Verde is that depending on the island where you are, you have different levels of admixture between Europeans and Africans. So there is a similar kind of context, very different environment, okay, it's not the Andes, but and very different culture probably, but you still have that interaction between the ancestral human genome and the strain of Helicobacter that may be present in a population or another, because also Helicobacter can be population specific, as long as I remember. So the short answer is I don't know enough, so I don't want to give an answer that doesn't really make sense. And I'm sorry if I'm a slightly odd position, but because I'm looking at you, really, even if you don't know. I'll try that. Maybe let me try that.
Thank you.
But I really suggest you to get in touch with Sandra. I can send you her contact if you want. So if Rafael puts me in contact with you, I can do that. I'm happy.
Thank you, Prof. Thanks so much. Yeah.
¿Alguna otra pregunta? Sin miedo, sin problema.
Buenos días.
Buenos días.
Por su presentación. Yo soy antropólogo y estoy haciendo una tesis de estudios de ancestralidad en nuestra población venezolana. Yo tengo dos preguntas básicamente. Uno que, bueno, la ancestralidad en algunos casos, tal como usted lo mencionó, puede usarse como un factor de riesgo para ciertas enfermedades, ¿no? Por lo menos esa es la tendencia. En el caso de poblaciones tan subestructuradas como las nuestras, es importante hacer un estudio de ancestralidad previo a los individuos que puedan presentar algún tipo para diagnosticar para ciertas enfermedades genéticas. O sea, ¿qué tan importante es hacer el estudio de ancestralidad previo en poblaciones subestructuradas como las nuestras? ¿Sí me copian?
So I can't understand the first part. So you're referring, so I understand the word centralidad, but probably I'm misunderstanding. I can't understand.
Ancestry.
Sorry,
Ancestry.
Oh, ancestry. Thank you. Sorry. Okay. So there are different things. So if you are talking about diseases that differ in prevalence between different ancestries, then having information about ancestry can give you a little bit of a help to predict the risk. So this is something that is already done in epidemiology, really, but you can kind of support it with genetic data. But then there are other things that are more, so I would say that in a population that is structured, what you want to do is that if you want to find the genetic behind the risk, you still have to do an ancestry analysis because you need to incorporate it. So it might not be necessary to do it to define risk, but you need to do it to understand the biology, basically.
Okay. Y la otra pregunta es, Últimamente, de un tiempo para acá, han surgido muchas empresas que hacen estudios de ancestralidad y otro tipo de análisis genético. Quería saber su opinión sobre este tipo de test, porque ya digamos que llegan a la casa una serie de información que muchas veces no estamos preparados para entenderla siquiera, ¿no?
Yeah.
Quería saber qué opinión le merece este tipo de...
Sorry, I didn't mean to interrupt. So yeah, there was the very famous case of 23andMe that was blocked for some time by the FDA in the US because on the ground that people are not ready to understand the results. So I think, so I have done 23andMe myself. So that's the one I can talk about. But of course, I've done it with a lot of consciousness. So there are two aspects to it. One is ancestry. So on the ancestry side, it's really hard to make people understand that there's no discrete groups between human beings. We are all a continuum, okay? And understanding where your genetics come from doesn't define you, okay? To me, my identity is not defined by my genetics. My identity is the language I speak, the culture I've grown up in. So it's very, so to me, it didn't change anything about my perception. Nevertheless, I came out 100% Italian, unsurprisingly. So, so it was very much in line with how I feel. So, so that's the ancestry. And then there is the disease related, which is, I think, what you're referring to the most. So they have, I can't remember how many of them. I think it's 4 variants, like BRCA, the genes associated with Alzheimer, and there are a few more for which there is a very, very strong, clear link between the genetics and the disease. And those variants have been, so you cannot access the information unless you click a couple of times saying, yes, I am aware of what this means, okay? So it doesn't pop up to you straight away. You have to say, okay, I really want to see this. What comes up to you straight away is some kind of indication towards traits and phenotypes, some of them relevant to disease. Okay, now I haven't looked at my results in a long time, but basically we're talking about eye color or earwax or caffeine metabolism, or, you know, you have a number of traits that you have some genetic evidence for. But then when you click on it, it does explain to you quite nicely that, you know, it's not predicting necessarily, it's giving you an indication. So I think I didn't see a problem in the way that they delivered the results. I think there is another problem, that is the way that genetics is perceived among people in the world, okay? There is a very deterministic perception of genetics. People think that if you've got the genetic variant, that's what is going to happen. And I think what we need to do, so as soon as I hear these things, when I am out with friends or family, I mean, family is a very good example, I think. I usually try and explain that there is a lot you can do with environment. I mean, the effects that we are explaining usually are only a very small fraction of the phenotypic variants still. So what we know from the genetic point of view is quite little compared to what the variance of the phenotype is. So I think it's really important. So I think the concern is real, and I think we need to be very careful about how messages get out there. For example, I don't know if that is something that you guys heard a lot about, but there were some studies about the genetics of educational attainment and social status in the UK. I don't know if that news arrived. That was really concerning, because it's like, well, wait, what are we saying? That you have a risk score for becoming good at school or bad? I mean, surely, yes, maybe there is some biological basis, but surely there is a lot you can do with the environment and the culture and the teaching and what the lifestyle choices. So I think it's really our job to try and pass the message as much as possible. I understand the concern, but to me, what I find more concerning is the fact that they make a lot of money out of people who want to understand their identity better. So all these companies started to tackle that need of understanding their identity of African Americans. Where am I from? Where did my ancestors came from? Where in Africa did they come from? I used to work in population genetics, received a lot of emails from people that they wanted to know where they are from. And it's like, how can I communicate to you that this is not going to change anything in your life? Because it's, first of all, we cannot tell you exactly where you're from. Second of all, you are where you're from. You speak the language you speak. You have the culture you have. I think these are social values that we are really forgetting, basically. So I think there is a concern. Frankly, I would hope that anybody who does a 23andMe test and finds something worrying goes and talks to their doctors and try and find more about what is going on. Another thing that doesn't come out at all is that there might be errors. So that is not a clinical diagnosis in any possible way. So you need to validate the result. So yes, maybe I'm too optimistic about humanity, but I would hope that people dig a bit more into their health issues before taking any decision based on a test that they paid $100. So, but yes, I understand your concern. I think it's a fair point.
Thank you.
Thank you.
¿Preguntas? ¿Suficientes? Sí. Well. Thank you, doctor.
Thank you.
Esperamos verla pronto.
I hope so. We should be in touch. Maybe we should organize a course.
Yes, yes, it's possible.
Yes.
I hope so in Costa Rica.
In Costa Rica?
See, when you go.
Well, I'm not planning to go to Costa Rica. I'm planning to go to Mexico again.
Okay.
Because I need to bring one of my students there.
Okay.
But yeah, we'll be in touch. We'll definitely be in touch, Rafael.
Yes. Bueno, muchas gracias por su tiempo, por esta magnífica conferencia y bueno, que haya sido del agrado y el entendimiento de todos y que bueno, nos dé unas luces sobre el estudio de GWAS, que siempre es un poco difícil de entender a primera hora. Bueno, gracias.
Solo quiero decir muchas gracias a ustedes. Lo siento que duró tanto y más de lo que esperamos por problemas técnicos. Si quieren estar en contacto, hay mi contacto Twitter en la primera slide. I'm happy to share the slides as well if people want to. Si tienen preguntas, si me quieren escribir, que lo hagan, por favor.